Hello, everyone, and welcome to Cure Talks. I'm Priya Menon, and today on Cure Talks, we are discussing how CRISPR-edited immune cells enhance fight against blood cancers. Joining me on Cure Talks on the Patient Advocates panel is Matt Goldman, Jennifer Abraham, and Maddie Hunter. The featured expert we have with us is Dr. Edward Stodmer, Director of the Bone Marrow and Stem Cell Transplant Program and Section Chief for Hematologic Malignancies at the University of Pennsylvania. Welcome to Cure Talks, Dr. Stodmer. Thank you for taking the time to join us today.
My pleasure.
Dr. Stodmer, I would like us to start with the most basic question for the audience. What is CRISPR? What does it do? And what is the excitement around it?
All right, we have 10 hours now to speak about this. Okay, no problem. So the most important thing to realize is that CRISPR is a biotechnology. It is a technology. It is not itself a therapy. It is just a way of engineering cells, and cells, are the basic components of our body and of our tissues. And when they're normal, they're working beautifully and doing things. And when they get all screwed up, they can sometimes cause abnormal proliferation, and become a cancer. Or they sometimes have genetic abnormalities in those cells so that they're not producing all of the proper things that they need to produce. And we, based on our knowledge of how cells work, based on data, over the last 50, 60, 70 years, all of the molecular biology and all the science that we have developed, starting with the discovery of DNA and how that is the code that makes these cells work, that has all led to our ability now to manipulate the genetics of these cells. And so CRISPR was discovered really over a decade ago, but was really sort of finessed by Nobel Prize winning scientists and it was first identified as a way, something that's very topical right now. Bacteria were infected by viruses. And CRISPR is the immune system of bacteria to basically chomp up viruses as they come into the cell. And the scientists who were studying this and found that whenever genetic material is chomped up, that right at the place where it is chomped up, number one, you can specifically find a place to chomp up, and then you can insert something into the place that is broken up, and that's really the basis of this biology. And the way genetic material is chomped up is an enzyme. Enzymes are the things that chomp things up. And so Cas9, we call it CRISPR-Cas9. Cas9 is an enzyme. And it's called an endonuclease, which is something that can actually chomp up genetic material, DNA or RNA. And but the way you guide it to exactly the gene that you want it to chomp up is you make little pieces of RNA called guide RNA. You insert that into this enzyme, and then you can make guide RNA to any gene you want to rip out or insert. And then you just mix cells in with this enzymatic process in basically a petri dish, and you can take out or insert genetic material into cells. So that's really the CRISPR technology is really a gene editing technology that allows you to insert or take out genetic material from cells. So So that's, and it was evolved from bacteria, and now human beings have been able to evolve it further to use it to try to manipulate anything that has genetic material.
Thank you. I think that was helpful. I remember in a couple of years back when Dr. June was here on this platform, Dr. Stodman, he was talking about CAR T cells and how cellular immunotherapy has revolutionized cancer treatment. Can you talk about these cells and the role of CRISPR-Cas9 in the use of CAR T for cancer treatments?
Right. Well, so the logical next step is, which cells do you want to genetically manipulate? And there's really, so far, there's only been two major types of cells that we've been trying to manipulate to try to help patients with diseases. The logical first step, though one of the more difficult steps, is to take a person who has a genetic disease, like sickle cell anemia, like thalassemia, like Huntington's chorea, and all of these. So the different genetic diseases. And then if you could, and these are genetic diseases that the patients inherited. So if you take out their stem cells, or their early cells, you can then take them out. You can use CRISPR to either insert the right gene in there or take out the bad gene. And then you can do sort of a bone marrow transplant and insert back in the cells that have been genetically manipulated, but now healed, and potentially cure a genetic illness. And the one disease that, or the two diseases that have been the most advanced in that, where human beings have actually gotten this, are sickle cell disease and thalassemia, with some very promising results. So that's one way of doing it. Those are called somatic cells, incidentally. And this is a really important point. Right now, we are comfortable with manipulating cells that don't have the capability of creating other human beings. We are uncomfortable right now with genetically manipulating embryos or germ cells, which will then which have the potential of curing or eliminating these diseases, but who knows what other consequences the genetic manipulation of germ cells will be. So and remember, that is something that was done once in China, where they were trying, I guess you could say, a noble goal of trying to keep people from getting HIV by making manipulations in the germ cell in embryos, but without any real good oversight, and who knows what things you're unleashing. So the germline manipulation has been something that we're still very hesitant to do. But the somatic cells are things that will not be inherited, will not alter the person. There's a misconception that when we use CRISPR technology, we are somehow squirting in something into a person and changing all of their genetics. And that is not what's happening. We are just taking cells outside of the body, genetically manipulating those cells, and it's those cells that we then put back into the body. So there's no CRISPR that we're putting back in. Now, the main thing that you were discussing, and our major area of research, has been the very revolutionizing therapy for many cancers, the CAR T cells, where we take the immune cells out of patients, engineer them, and we've been engineering them for decades now, where we insert genetic material into them to put on their surface warheads, and then grow up those cells and then give those cells back to the patient, where they go to the cancer. These are warheads that are directed against the cancer to destroy the cells. And these it's been tremendously successful for blood cancers, particularly acute lymphocytic leukemia and lymphoma, and just recently multiple myeloma, where we see tremendous responses. But it's not everybody. There is still some real toxicity to it. And many of the patients, despite having beautiful responses, will still relapse the disease. So CRISPR technology is being harnessed to try to see if we can improve those CAR T cells. And that's really the main thing we've been doing. And we've been doing that by taking out those same T cells, doing the same thing of infecting them actually with a virus to insert into them the genes to put on their surface the warheads. But we're also using the CRISPR technology to take out certain genes that we think inhibit their function to make them even more active and more directed against the cancer. So that's been one major way that we've been using it. And in fact, the study that we did was the first study to insert any CRISPR manipulated cell into a person, but it was particularly these sort of CAR T cell like cells that had been manipulated to make them to hopefully work better and proliferate and stay in the system better.
This is very interesting, Dr. Studmar. I'm going to get the panel to join the discussion now. We have Matt Goldman. Matt is a myeloma survivor and advocate. Matt writes about his myeloma journey on his blog, Matt to Assess Myeloma. Matt, you're on.
Thanks, Priya. And thanks, Doctor. With CAR T, there's this period of time where your T cells are being manufactured. Is there going to be sort of a similar manufacturing period for CRISPR when you're editing the genes? And does that mean that folks maybe who are a little bit more, a blood cancer patient who maybe is a little more critical or not stable might not be eligible for this?
Well, you're exactly right. The biggest, if there's such a thing as the biggest problem with CAR T cells, is that the patients that we're treating are in general patients who have very advanced disease, and there aren't that many other options for therapy. And so their disease is growing, and then you collect their cells, and it can take a month to 6 weeks to engineer them, because you have to first process them, put on their surface the warhead, and then they have to grow up in their system. And then there's a whole process of making sure that we call them release criteria, that they're safe and they don't have infection in them, and that they're going to be safe for the patient. And you're right. So you have to figure out how, the term we use is to bridge the patient for potentially 6 weeks, which can be very difficult for patients who have very advanced disease and don't really have other options for therapy. And to tell you the truth, this is exactly the exciting area of CRISPR technology, because when you use your own cells, the autologous cells, you have to go through this process. But what if you could use someone else's cells and cook them up well before they're ever needed, put them on the shelf, and then you're ready with either lymphoma-directed CAR T cells or a leukemia CAR T cells or myeloma CAR T cells. And then when the doctor and the patient make the decision that it's time to get this treatment, next week you can get the treatment. And so the problem, though, of course, with donor T cells is they're not the same as you. And so your body so there's a number of hazards. Number one, your body sees them as foreign and potentially will reject them. Or number two, those cells will see you as foreign and attack you, and not just the tumor cells. So we can use, and those are, of course, the way that your body recognizes the cells, or the way those cells recognize your body, are based on proteins on their surface. So if you can CRISPR edit them and take out those proteins, that identify that make them damaging to you or make them identifiable to your immune system, so that they really are, that your body is blind to them, then they can be squirted in there and do everything they need to do. And that's exactly the number one way that we're using CRISPR right now. We're taking healthy donors, we are CRISPRing out the genes that identify that lead to compatibility, and still making them into putting warheads on them, and those studies are ongoing right now, where we're trying to use autologous allogeneic, we call them, donor cells. So I think that in many ways CRISPR is the route to fix the problem that you've identified.
Okay, well, you just addressed my second question. I was going to ask about the off the shelf.
Yeah, no, off the shelf. I mean, it is our goal. Now, I must admit that we are also working on making the processing of autologous cells much quicker. And in fact, at our center, we have a procedure that we think might even make these cells from harvesting to ready to go in less than a week. And that would be ideal if you can harvest someone on Monday, infuse them on Friday, then you could do that. But this approach, using the CRISPR technology, given that, and really it was our study that demonstrated, the first question, of course, is this safe? And is this feasible to actually give these cells to patients? And fortunately, that was really the main thing that our study showed, was that it did appear safe. There was no danger or untoward toxicity to infusing these cells, and they survived in the patients. They didn't just immediately be eliminated. But now most of the CRISPR work is directed on trying to make these off-the-shelf products.
That's amazing. The last question for me is, it seems like we're seeing with CAR T and myeloma patients, we're not necessarily getting that durability that maybe the other blood cancers are getting. Is this an issue that maybe CRISPR can work through, or is there still some unknown about myeloma that makes it a little more challenging?
Well, no, you're and definitely this is, well, so what we have seen in ALL in particular, in the acute lymphocytic leukemia in children, is persistence of these cells in the body really does seem to correlate with success, with long-term success. In fact, that we've coined the term, they're serial killers, right? Like that they do their job, they then go to very low levels, but stay in the body. And then if there's any hint of the disease starting to come back, that stimulates the CAR T cells to proliferate and do their thing. And that seems to be really evident in ALL. It's less so, but pretty evident in non-Hodgkin's lymphoma. But you're right, in myeloma, there has been the persistence of these cells does seem to be less. And and though we in fact, my personal longest duration of a patient in remission after CAR T cell is 4 years and counting. In the average person who has gone into remission with CAR T cells does show signs of progression within a couple of years. And the term we use is there doesn't appear to be a plateau to the curve yet, that it looks like if you wait long enough, the majority of people will have their disease progress. So this is the major focus of research right now, is how do we overcome that? How do we make it so that these cells will truly kill every last drop of myeloma and keep it from coming back. And there's so many different strategies. I mean, strategies of using earlier cells, healthier, younger cells that aren't as exhausted, using different, right now, remember the number one target in leukemia and lymphoma is what we call CD19. That seems to be on the surface of most of the lymphoma tumor cells. In myeloma, the target is called BCMA, the B cell maturation antigen, which is a target on virtually all plasma cells and not too many other cells. So that's what makes it safe. But maybe, and BCMA has certainly been very, or BCMA-directed CAR T cells have been very effective at killing myeloma cells, but it seems like it doesn't get rid of all of them. So number one, there's different ways of engineering the warhead against BCMA. There's other targets on myeloma cells, just like, remember, daratumumab is a monoclonal antibody, and elotuzumab is a monoclonal antibody, and they're directed against CD38 and CS1 or SLAMF7. And so they've made CAR T cells against those things. And maybe some combinations of these different targeted cells might ultimately lead to a better result. But then ultimately, CRISPR, which is the topic of today, is you can use CRISPR. And if there's anything that our study suggested was that by CRISPR, so there's a gene, or there's a whole mechanism of that inhibits the activity of immune cells. We call it PD-1, and we've learned that if you inhibit PD-1 on these cells, those cells can really proliferate even more. And it's led to the whole another area of immunotherapy that has been very successful in other cancers, not as successful in myeloma, these PD-1 inhibitors that you see advertised on television all the time. And well, one of the things that we did with our CRISPR study, and what a lot of these studies are doing, is we can genetically eliminate PD-1. So in effect, you don't have to add that treatment, a PD-1 inhibitor. In effect, it's gone already, which will lead to the proliferation and expansion of these T cells. And that was what we showed, that in our study, contrary to what we sort of had observed, that the cells that were CRISPR edited to remove PD-1 seemed to stay around in the patient's system for, as far as we studied, up to 9 months or maybe even a year, where most of the time, most of the BCMA-directed CAR T cell studies have shown that within 3 months, most of the CAR T cells have disappeared. So CRISPR editing certain genes that lead to the proliferation of these cells may be very helpful.
Wonderful. That's all I have.
Thank you, Matt.
Thank you so much, Doctor.
And next on the panel,
my pleasure.
Yeah. Next on the panel, we have Jennifer Abraham. Jennifer is a cancer warrior who is part of a melanoma, thyroid cancer, and CLL. And she's in complete remission now. Jennifer, please ask your questions.
We got to clone your genes and figure out how to fight off all of those things. That's great.
Yes. Well, thank you. Thank you so much, Dr. Stadmeyer, for being here, and for, this is amazing. And your explanation was wonderful, because I've been trying to really educate myself on CRISPR and having a little bit of difficulty. But I actually have, so along with my questions, I have a question that just came up during this, which I hope it's okay if I ask. But
Sure.
So the first thing I thought about is, I had my melanoma at 20 years ago, thyroid cancer at 18 years ago, both during the pregnancies of my children, and then CLL 8 years ago. Right after the diagnosis of the CLL, we did the Prognostic sequencing or which, and I had all the bad prognostic factors. But we knew that 8 years ago, and it was a matter of watch and wait for 4 years until I had severe disease progression. But would there be a way at diagnosis to take my cells? I mean, if I had an option, could you take some of my healthy cells, put them on a shelf, and keep them, and then if I needed this CRISPR, I mean, if that was an opportunity for me now, if it was an option for my children,
Sure.
just given my history, I'd be giving you cells like crazy.
Sure. No, I think that's a great question, and a question that does come up frequently. I would say right now the whole area and the whole science of this is evolving so quickly. It's premature to basically say, why don't we take the entire population and store away T cells for a rainy day? It sort of reminds me a little bit of the umbilical cords. Like, should we store away umbilical cords? And now we've learned, unfortunately, that umbilical cord stem cells were not the answer to all of our prayers, and so they really are not utilized much at all. But it's wonderful that there is a registry of those things. I'm very, very optimistic that we're gonna have these vats of cells that have been already engineered for various purposes and will work very well, that probably we won't need the personalized T cells to be stored away. And you could always argue that a person, especially if somebody has a propensity to develop a disease, maybe their immune cells are not going to be the most healthy, because you could argue that maybe that's part of the reason why the disease occurred in the first place, right? Remember, our body is constantly being surveilled by our own cells. In fact, it is the T cells that are the most anti-cancer cells in our body, and so if a disease is created in the environment of your own immune system, then that might suggest that the immune system, it might be better to get somebody else's cells. So as of this moment, I don't think I would rush to store. I haven't, I remember my son is now 26 years old, but 26 years ago, the big question was, should I save his umbilical cord? And I decided I was going to live by my own rules, and I did not store his umbilical cord. And so far, we're doing okay. Anyway, but a very good question, an important question.
Thank you. I actually did store both of my girls in stem cells. Yes. The other question is, given my other two cancers, when I was diagnosed with CLL, it actually became really difficult for doctors to know what to do with me. So I kind of had to acquire a team, and one of them said I was a medical anomaly. And at the time of diagnosis, there was actually nothing that they could recommend, because they could have caused progression of other cancers or brought on the other cancers. So given that, but I have responded really well to, I've been on a clinical trial, but given that information of these other cancers, and now I worry about developing other cancers, how would CRISPR, because I know I'm not eligible for CAR T, so how would CRISPR be in terms of other cancers? How does it work?
Yeah. Well,
how does it target all the other possibilities?
No, I think that's a great question. And yeah, so that was sort of my point that at the beginning is, remember, CRISPR is just a technology. It is not a therapy unto itself. It is just a way of manipulating the important disease fighting cells of our body to do their thing better. But there's a whole area that I must admit I'm not an expert in. of CRISPR diagnostics. So CRISPR technology has actually been used to identify the coronavirus and identify the various strains of it, and so because it's looking at genetics of this, and so certainly. And it's a very sensitive sort of testing. So one of the potential uses is not so much in therapeutics, but sort of, especially in your body, it would be interesting to see both the abnormal cells and the normal cells, and whether there are any predictors. Are there certain mutations that are there that potentially, if you could suppress them or change their natural history, that would be good. And you could see, and so potentially CRISPR technology can help with those diagnostics, those prognosticators, et cetera. But in terms of actually treating disease, I'm very confident that we will be able to figure out other cells to manipulate. But right now, the number one cell that we, because it is the most important cancer fighting cell, are those immune cells, those T cells. Interestingly, there's probably like 30 different types of T cells, and more we're trying to identify which subset of those or particular cells that we should target the most, and that's another way that we're trying to enhance the therapeutics for use with CRISPR.
Thank you. My other question is, I'm very active in the CLL community as a patient advocate, and I have never heard of or read about really in CRISPR as being an option for treatment of CLL. And how do you see that moving forward?
Well, I'll tell you
for myeloma.
Yeah, sure. Well, the first disease actually, so in like 2000 was it 2010? 2010, the first the first CAR T cell therapy in human beings was CLL, and we did it with and 2 of the 3 patients that were presented in the New England Journal of Medicine are still alive and well today. So CLL, but interestingly, none of the products have yet been FDA approved for CLL. I think that's about to change. So I think there's a lot of, and I think that it really is an area where enhancing the activity of the CD19 directed CAR T cells will, with CRISPR potentially, will really enhance the likelihood that patients with CLL will improve with that therapy.
Thank you. Thank you very much.
My pleasure.
Thank you, Jennifer. Our next panelist is Maddie Hunter. Maddie has been living with myeloma for past 20 years, and she leads the Philadelphia Multiple Myeloma Networking Group. Maddie.
And Maddie has taught me most of what I know about myeloma.
Oh, it's great to see you again, Dr. Statmauer. In the life of the Philadelphia support group, we have not talked about CRISPR. Nobody's brought it up. And to your point about it being a technology, and we're, I think in the life of the average patient, the interest is in treatment, not in necessarily what's behind the scenes. But so I'm fascinated with all of this. I've learned a lot just in the last 30 minutes. So I'm scrolling through the questions that I had thought to ask. Some have been answered. So in terms of the application to myeloma, many of the folks that we have in our group have MGUS or smoldering myeloma, those precursor states. So what's the relevance of CRISPR potentially to that group of myeloma patients who are not actively being treated at the moment?
Yeah, well, I think that's a really interesting question. And in fact, the precursor states, MGUS and smoldering myeloma, are probably the number one area of focus right now in the myeloma community. it's obvious that if we can, we now identify patients sometimes decades before they actually develop active myeloma. And our current standard of care is to watch them. And wouldn't it be great if we could just eliminate the disease right then with some less toxic and very effective sort of therapy? And that's the conundrum, that every time we think, and there are many studies that have shown that treating patients early with smoldering myeloma can lead to longer responses and even improve survival. But it's really tough to balance that with somebody who has the potential of being alive and well 30 years before they ever need any therapy, and treating them then and giving the toxicity and the side effects of therapy. So number one, again, using the CRISPR diagnostics might be very helpful in identifying better, even the prognosticating the people who are imminently going to evolve into more aggressive disease and potentially treating them early versus those who won't. And I think CRISPR technology has the potential for that. But also, the more we work on making the cellular therapies less toxic and more effective, that will, of course, then we will be thinking of using them earlier and earlier in the course of the disease.
Interesting. Sort of following on, you may know that my dad also had myeloma. My brother was just recently diagnosed with MGUS. So curious about the relationship of CRISPR to some of these more familial patterns. I know the PROMISE study is trying to help us understand the genetics of the disease, but what do you see as the possible application?
Well, the bigger picture of the genetic basis of myeloma is a very important and fascinating area. And so unlike diseases like we talked about, thalassemia and sickle cell disease and cystic fibrosis, where you can identify one gene and it's a screwed up gene and the product of that gene that leads to the disease, and so you can target that gene and make things better. There has not been one gene in any of the diseases we talked about, CLL, in myeloma, in lymphoma, in ALL, that is driving the entire disease, and that's the problem. Every time I go to the International Myeloma Foundation meetings, Brian Dury will frequently raise his hand, will say, raise your hand if you have a first-degree relative who also has myeloma or something. And where, it is a rare, officially myeloma is an orphan disease, a rare disease. And yet in those rooms, we usually see 5%, maybe even 10% of the participants raise their hand. So there's no question that there is something to this, that there's some familial thing. Now, of course, there's always the question, is it the environment? Families tend to live in the same area, and is it the environment and that's causing it, or is there truly some genetic aspect to this? And it's probably a combination of the both. But as of this moment, it doesn't seem that is as targetable as some of the other diseases that have just one genetic abnormality, or at least a primary driving abnormality, like breast cancer, the BRCA gene and things like that.
Oh.
Yeah. So what are some of the challenges of using a tool such as CRISPR?
Well, so whenever you use something that chomps up genetic material, you got to make sure it doesn't chomp up good genetic material or reconnect genetic material in a way that will make those cells grow abnormally, and in fact, that is part of every study we do, is looking for we call them translocations and genetic breaks. And number one, are these CRISPR edited cells having other breaks that are in known cancer causing genes, so that you might have a cell that will now become cancerous because of the genetic manipulation? Or is it in other genes that can make the cells die and not function that well? The good news, though this is still early investigation, is that it has been very, very, very rare that the translocations have been in any genes that are really concerning genes. So that's good. Number two, by the time the manufacturing process has been completed, and incidentally, the CRISPR editing occurs right during the same process. It's like a day or two process. While you remember, the process of creating these cells is to take them out, is to activate them, is to infect them with viral material to put on their surface the warheads, and to incubate them in CRISPR-Cas9, the enzyme, to chomp up the things. And that all happens really within a couple of days. So the real action occurs in a couple of days, but then those cells are incubated and grown up and then tested to make sure that they are safe. So what we have found is, by the end of the process, let's say it takes 10 days to do this whole process, by the end of the process, very, very, very few of the cells that are still there have any of these abnormal translocations or gene breaks that we didn't intend to make. And when you squirt them into patients and you actually then take these living cells that are in the patients out by drawing them and testing them, you basically see none of those translocations. So far, that whole process has been very safe, and but it's going to be something constant. And I don't know if you realize this, but the FDA mandates that any patient that gets a genetically infused manipulated cell, those patients on clinical trials have to be monitored for 15 years to make sure that there aren't any untoward craziness that happens. And so, knock on Formica here, so far things have been just fine.
I have lots more questions, but I'll ask one more and then look to you, Priya, if there's time for another. So we're fortunate that Penn has the capability to be involved with the technology. How ubiquitous is the capacity to use CRISPR technology across the country, across the world?
Yeah, well, most of these studies are being done by industry. So there are, in fact, there's a company that was started by one of the Nobel Prize winners called CRISPR Therapeutics, and it's one of its first studies is in multiple myeloma, utilizing cells that have been CRISPR edited so that they're donor lymphocytes and are able to be used for the treatment of myeloma. So I would say that that's probably the best way of getting access to throughout the country and potentially throughout the world, is that industry has found it to be interesting, and there are numerous sites within the country and also throughout the world that are studying this using. And so you only need to have a central manufacturing area, like one lab that is is fully accredited for this sort of activity. And then there's a number of companies throughout the world that are trying to utilize this technology to make therapeutics will be, we're fortunate at Penn is that we have our own capability of doing some of these pilot studies, but certainly even if the results are very promising, ultimately you need a partnership between industry and the academic centers to really make it available to the full population in need.
Well, so that just leads me to one last follow-up. So if industry's involved, that would suggest to me that the horizon for something very much more ubiquitous in the world is on a short horizon. Like they're going to look for a turnaround in their ability to make money off of this technology. So would you say the promise of the use of CRISPR technology is right around the corner for the myeloma world?
There are studies going on right now, and not only in myeloma, but in numerous other blood cancers, to do this. Just as a real aside, one of the biggest problems of CAR T-cell therapy in blood cancers has been the myeloid diseases, acute myeloid leukemia, and it's because the tumor cells, the leukemia cells, look very much like bone marrow cells, like normal bone marrow cells. So if the CAR T cell is successful at killing the tumor, it tends to kill the bone marrow. And so you have to have a donor bone marrow transplant as part of the CAR T cell therapy, which sort of almost negates the benefit, Yeah. So they've been using CRISPR technology to actually make a person's stem cells resistant to the CAR T cells, and therefore are able to now give the CAR T cells that will kill the leukemia without killing the bone marrow. And so there's a real so CRISPR technology for some of the, even for other, so many blood cancers will be potentially very, very helpful. And I think these clinical trials, it is very rapid. These clinical trials are ongoing, and so we could even potentially see within a year or 2 years some real therapeutics available for our patients.
Thank you, Dr. Stadtmauer.
Thank you, Maddie. Dr. Stadtmauer, I have just one last question before we wrap up. How do these, can CRISPR-edited CAR T cells, help with solid tumors as well?
Yeah, boy, solid tumors have been the like the holy grail, have been really, we'd love to do it. It has not been as easy. It hasn't been, it's a tougher nut to crack, as we say. Sometimes I think of it, a solid tumor, as an onion, where like blood cancers, there's blood flowing through them. And so the CAR T cells have easy access to the tumor cells. But in a solid tumor, you basically have this golf ball, and the CAR T cells are able to sort of peel off a layer of the onion, but there's still a big ball of tumor that doesn't seem to be as amenable to the therapy. So I think CRISPR technology has a tremendous, or engineering of these cells has a tremendous potential for finding ways of getting through the onion and peeling it down and getting rid of those tumor cells.
Thank you, Dr. Stadtmauer. Thank you for your time today. I think it was great talking about CRISPR and how this gene editing tool has the scope to help with cancer treatments. So thank you very much. Matt, Jennifer, and Maddie, thank you for your participation and all the great questions. This talk will be made available on the Cure Talks website and also on the University of Pennsylvania's pages. Thank you, everyone, and have a great day.